Hi, good morning, everyone. Thank you, Gabriel, for that introduction. And thank you, everyone, for joining. So as Gabriel introduced, we're here to talk about chickens, the meat chicken and specifically. And here it is. The modern broiler, also known as a meat type bird, is one of the results of decades of research in genetics, nutrition, and husbandry. And it's resulted in its upward trajectory in terms of its efficiency, being able to produce 2 to 3 kilograms in weight within 4 to 6 weeks of age and under less feed than ever before. And that's that efficiency that's also led to its rise in affordability, making it one of the most affordable forms of animal protein on the market. Animal protein also being a high quality form of protein. And all of these have also contributed to its popularity. Consumers tend to prefer chicken over other forms of animal protein, not only due to its affordability, but also because its connection to healthy diets, as chicken meat is lower in saturated fats. And it's often included in healthy diets that are popular among mainstream and upcoming individuals. However, how do we continue this upward trajectory of chicken? Us as an industry and as researchers need to ask us this question, as it is within our means to be able to continue to provide animal protein affordably, efficiently to those that want it. The first thing I'm going to say that we need to focus on is robustness. This includes the animal's welfare, its overall functioning, and its health, as well as its sustainability. In a world where resources are becoming more and more limited, as well as populations increasing, especially in areas like Africa and China, where the demand for meat and animal protein continues to rise, means that we need to be able to produce our birds in a sustainable fashion. And finally, the welfare of the animal, which should not only be a concern for us due to the fact that more and more consumers are starting to care about how the welfare of the animal is cared for throughout its life, but also as stewards of animal protein animals, we should care as well. The majority of the growth of the broiler that is the efficient, makes it the efficient form of animal protein across the world actually occurs within the long bones, where from day 1 to day 42 of age, the long bones increase approximately 4 times in length and 7 times in diameter. This occurs at the growth plates through a means of dynamic molecular reorganization known as endochondral ossification. However, this fast growth rate has led to specific points within the skeletal system that have been shown to have wound sites and diseases such as bacterial chondronecrosis, osteomyelitis, what we're here to discuss today. These regions are first in the spinal cord, where things such as kinky back are seen to occur most frequently, as well as the proximal femur head and the proximal tibial head. It's at these key stress points within the skeletal system that wound sites and diseases such as bacterial chondronecrosis, osteomyelitis, or femur head necrosis are found. The bacteria that leads to these infections can enter into this bird via either the respiratory tract or the intestinal lining, either through stress-induced immunosuppression and damage to the epithelial lining, or due to stressors in the bird, or overload of pathogenic bacteria able to then cross that epithelial lining and enter into the bloodstream. Once there, they can enter into the bone via the highly vascularized growth plate, where, unlike mammals, avians have extensive vasculature all the way through the entire proliferating zone. At this growth plate in the mechanical stress points where wound sites have occurred, there is exposed collagen where bacteria are able to induce infection, leading to things like osteomyelitis that we see in BCO. Looking at the growth plate more closely, as shown diagrammed here on the left and in an image on the right in the proximal tibial head, there are 4 main regions. The first being the epiphyseal or the articular cartilage cap, the white cap shown on top of the growth plate region. Then the metaphyseal region, followed by the diaphyseal region where the actual bone is being laid. Within the metaphyseal and growth plate region, these cells depicted here are chondrocyte cells, which undergo several different stages in cellular growth and proliferation, as well as final hypertrophy, in order to create the matrix needed for bone to be laid. These regions could also be seen very easily visually within the bone. Also depicted in this diagram, the vasculature can be shown to come both through the articular cartilage cap down into the growth plate, as well as up from the diaphyseal region. It's at this junction between the hard articular cartilage cap and the highly dynamic growing growth plate that we see the high susceptibility of BCO due to the induction of the occurrence of wound sites here. These wound sites can actually be seen under histological examination, as shown by the arrows, where that hard articular cartilage cap comes into contact with the highly dynamic growth plate. These wound sites are evidenced by chondronecrosis and exposed collagen, and often can transect this vasculature that comes through both regions in the growth plate, as shown here from the blood vesicles here. This creates two things. One, it allows for direct access of any circulating bacterial species to come into direct contact with necrotic cells and exposed collagen, for then collagen binding to occur. Second, it also creates an hypoxic environment, as the crossing and transecting of vasculature limits the capacity of blood flow in other regions. These two things are extremely conducive for bacterial colonies to then lead to what we see as BCO or FHN, in the proximal femur head on the top and the proximal tibial head on the bottom, starting with a clean articular cartilage cap, macroscopically normal femur, and following the degrees of severity. BCO is phenotypically shown to have inflammation, bone attrition, and bacterial infection, with even the production of biofilm in severe cases. And finally, understandably so, this type of necrosis and infection can lead to a healthy broiler becoming bacterially BCO and induced lameness. That lame broiler that we often see in concerned images is, for one, a welfare concern, as broilers undergoing lameness have been shown to access feed with painkillers, preferably over feed without. It's also a food safety concern, as this form of lameness is bacterially derived. And finally, it's an economic concern, where lameness can decrease performance as well as decrease the overall number of birds our growers can take to processing. So while there is a theory and understanding of how bacteria can enter into the bloodstream and access the bone within BCO, and we have the phenotype of necrosis, bone attrition, and inflammation, what has yet to be fully elucidated and was the focus of my research is how bacteria directly cause BCO. Here in the DREEDY lab, we are a molecular genetic and endocrinology lab focusing on cellular mechanisms. To that end, we're able to use specific known BCO isolate, S. agnetus 908, Staphylococcus agnetus 908, which was a strain isolated from flocks with BCO here at the University of Arkansas. And before I started my PhD, our lab found that dicer-1 dysregulation under Staphylococcus agnetus 908 infection and in BCO-induced femurs led to an accumulation of double-strand RNA, activation of NLRP3 inflammasome, as well as accumulation of IL-1 beta, which decreased cell viability in bone cells and understandably would contribute to bone attrition seen in BCO. This will be described in more detail here soon, but it was this foundational research that then led me to focus my attention on the mitochondrial dysfunction potentially present within bone cells under BCO conditions, as well as autophagy dysregulation. And finally, looking at the pro-inflammatory factors that could be contributing at a systemic level and the local level in BCO-affected broilers. So starting with our experimental design, all of the samples used in our studies were from either a wire flooring model, as shown here, developed by Dr. Weidman at the University of Arkansas, where healthy individuals were taken from birds raised on litter flooring, shown in the back, under normal conditions that were non-lame. And BCO-affected samples were taken from the femur and tibia from lame birds reared on wire flooring with severe BCO lesions in both legs. Blood samples were also taken before euthanasia. Here we have a depiction of the bone severity scores we took. So healthy bone was our normal bone on the left from a non-lame bird, and severe BCO necrosis on the right from a lame bird. For our in vitro studies, we used human fetal osteoblast cells from ATCC for both Assignetus 908 challenges, as well as cellular pathway manipulations and cytokine and chemokine exposure. Primary chondrocytes, which will be discussed in more detail later on, were also used. Sample preparation analysis, I won't spend too much time here. However, gene expression was analyzed via real-time qPCR, protein expression using Western blot analyses. There was also immunofluorescence on primary chondrocyte cells, with MTT viability also being used for cellular viability assays. Starting off with double-strand RNA, the QR code shown on any image depicting the publications of our research will take you directly to these publications to be able to read it more thoroughly. So in this study, we starting here on the left, we actually had both human as well as chicken bone tissue, human osteomyelitis and chicken BCO. In both, we found downregulation of both protein and gene expression of dicer1. Dicer, under homeostatic conditions, almost acts like a pair of scissors, cleaving double-strand RNA within the cytoplasm for further processing. This mechanism is essential for maintaining cellular homeostasis and also important in immune response pathways. So there was a commonality between human and chicken tissue with dicer1 dysregulation. This knockdown in dicer1 also was coupled with an accumulation of double-strand RNA, as shown here, in both BCO and human osteomyelitis samples. Under an in vitro model, we saw the same pattern where treatment with Assignetus 908 led to an accumulation of double-strand RNA, as seen here, as well as knockdown of dicer. Further investigations of protein expression under our in vitro model showed that LNRP3 inflammasome was activated under Assignetus 908, as well as caspase-1, and that knockout of dicer congruently also activated these inflammasome pathways. These pathways are often activated in cell death pathways, and with LNRP3 and caspase-1 being key regulators of them. All of these also coincided with an accumulation of IL-1 beta, a potent pro-inflammatory cytokine, all contributing to the potential of this pathway being activated under BCO conditions and leading to bone attrition. Next, we looked at mitochondrial dysfunction. But why? Several reasons, starting with the bacterial component of BCO. Bacteria have been known to hijack mitochondria not only for the production of their own ATP, but also for dysregulation of cell homeostasis and abilities. Specifically, certain bacterial species known to cause BCO have been known to target mitochondria via active formation of pores, disrupting the mitochondrial membrane potentials and wreaking havoc and cytochrome C activation and cell death pathways. Additionally, in the bone in general, mitochondrial functioning is key for regulating the ratio of bone forming to bone absorbing cells like osteoblasts and osteoclasts, as well as that ratio then disrupting the formation and resorption seen in aging and disease. Mitochondrial dysfunction is also incredibly important for maintaining decreased oxidative stress and structural components of the bone. To that end, we hypothesized that mitochondrial dysfunction could be involved in the BCO pathogenesis and published our findings in 2021. Starting with looking at genes regulating the mitochondrial biogenesis in both normal tissue in blue and on the left, and BCO-affected tissue on the right, biogenesis was shown to be upregulated via upregulation of D-loop and PGC1 alpha and 1 beta. When looking at mitochondrial dynamics, meaning the balance of fission and fusion within mitochondria, we saw that OPA1 was significantly downregulated, while MFN2 was upregulated. Looking in more detail, OPA1 is essential for intermitochondrial fusion when fusion occurs. Mitochondrial fusion is often seen in stress states as the increased network capacity allows for increased energy production and processes needed. In the case of fission, Fission often occurs when mitochondria needs to be processed via mitophagy, or there is damaged mitochondria, or as a stress response. Mouse studies have shown that this decreased OPA expression can lead to fractionation of mitochondria and breakdown of mitochondria, as well as increased cell death, which was able to be mitigated through overexpressing of OPA1. Finally, looking at mitochondrial functioning genes, we saw that several genes were downregulated under BCO conditions, starting with ANT, COX5A, COX4, indicating a decreased expression of the machinery necessary for mitochondria production of energy. Also indicating disruption of the function was avian UCP mRNA expression being significantly up, with avian UCP acting similarly, avian UCP being significantly upregulated, indicating that the ATP production could be being dysregulated and conversely being used for heat production rather than ATP production as protons move across the inner membrane. So in conclusion, this is the first study to implicate mitochondrial dysfunction in BCO conditions, which could be potentially the cause for increased cell death and chondronecrosis, as well as alteration of cellular processes, and be contributing to previously found mechanisms of double-stranded RNA accumulation. However, further research is needed. A major limitation in this study was the lack of protein expression analysis and mechanistic underpinnings. Next, we sought to determine if autophagy dysregulation played a role in BCO. We published in 2022 in Poultry Science, which you can find here using the QR code. Why autophagy? Well, while autophagy is a cellular process that uses autophagism and lysosomal degradation to not only rid the cell of foreign bodies, but also damaged organelles, it also can play a dual role in cell survival, where under bacterial infection, it is one of the key mechanisms in which the cell can rid itself of bacteria. However, certain species, one in particular being Staphylococcus aureus, which is a known causative agent in BCO and osteomyelitis in humans as well, has been shown to hijack the autophagy pathway, turning it from a cell survival to a cell death mechanism. Specifically, S. aureus will block the fusion of lysosome to the autophagosome, allowing it to proliferate within the cell under the protection of the membrane before then continuing its infection. Also, at homeostatic levels, autophagy is integral for bone mineralization, as well as the regulation of bone cells ratios of osteoblast to osteoclast, and the management of reactive oxidative species produced under bone homeostatic functions. Starting with the tissue and looking at the initiation stage of autophagy, we saw that Beclin-1 protein expression was significantly upregulated, while ATG13 was significantly down. Beclin-1 gene expression was also measured without significant differences. The nucleation stage of autophagy was also investigated, with HG5 protein expression showing no significant differences. However, HG14 gene expression was significantly down in BCO. Elongation protein expression showed a significant decrease of LC3 under BCO conditions. mRNA expression of key factors in the elongation stage of autophagy also found LC3C mRNA expression significantly decreased, as well as ATG9B, ATG16L, ATG10, ATG4B, ATG2B, and ATG12. The other genes listed down here were also analyzed without significant differences. And lastly, the fusion stage of autophagy in BCO tissue, where protein expression of Rab7 showed no significant difference, its mRNA expression was significantly decreased, along with SQSTM1 or P62. An in vitro study was then undertaken using S. ignitis 908 on HFOB cells at a multiplicity of infection of 50 to 1 for 24 hours. First off, no surprise, showing a significant decrease in cell viability under challenge with a known BCO isolate. We then sought to determine the state of the autophagy machinery from these cells challenged with BCO isolate S-ignitis 908. Protein expression showed decreased in many of the machinery, however, with a significant increase in LC3 type 1 to type 2 to type 1 ratio. This bears weight in the fact that LC3 type 2 is converted to type 1, which type 2 being membrane bound, type 1 being cytoplasmic. And this is essential for the formation and lengthening of the autophagosome for then fusion with the lysosome. When this ratio increases, it shows a lack of autophagy completion, potentially. There's many different ways to inhibit autophagy. However, we focused on some specific inhibitors that block autophagy at 2 different stages. First, being in the early stages of autophagy, we use 3-methyladeline, and then later blocking the fusion of the lysosome to the autophagosome using chloroquine. To determine if autophagy could be the culpable factor inducing the decreased cell viability, we used both these inhibitors, and both showed a significant decrease in cell viability comparable to that under bacterial infection. The autophagy machinery was also understandably dysregulated under these inhibitors, with again seeing that significant increase in LC3 type 1 to type 2 to type 1 ratio. In summary, there's several stages along the autophagy pathway that were shown to be dysregulated, both in BCO-affected tissue, as well as in cells challenged with known BCO isolates. These evidence is indicative of decreased autophagy within BCO tissue, and when challenged with a known isolate of BCO, this dysregulation continued alongside decreased cell viability. And inhibition of autophagy via separate mechanisms results in not only dysregulated autophagy machinery, as seen in tissue and bacterially challenged cells, but also decreased cell viability overall. So the dysregulation of autophagy in BCO could be caused by the bacterial manipulation and could be contributing to the BCO pathology by affecting cell survival and homeostasis. So both these pathways are within the bone tissue itself. However, it's clear that bacterial infections can cause systemic effects within the broiler, and that these effects could potentially be measured within the circulation as potential biomarkers for this disease. To that end, we published in 2021 looking at the local and systemic cytokine, chemokine, and FGF profile in BCO. Starting with the plasma, wanting to determine if there was a systemic effect that could be altering bone cell function, we treated HFOB cells and primary chondrocyte cells with plasma from both BCO and healthy individual broilers, with plasma from BCO broilers leading to a significant decrease in cell viability, indicating something within circulation affecting bone cell function. To determine what that could be, we analyzed the cytokine profile within both the bone, local bone, and the circulation in whole blood. Bone had a very clear profile of IL-1 beta being significantly upregulated, as we had shown in previous research. Blood had several factors, including TNF-alpha and IL-12B significantly upregulated, with TNF-alpha being a potent pro-inflammatory cytokine. We also looked at the chemokine profile, where bone and blood both had IL-8 significantly upregulated. Blood also had CCL20 significantly upregulated. IL-8, while avian has 2 orthologs, they are 56% identical to one another, and they are most closely related, identical to CXCL8 in humans. Most notably, IL-8 is involved in migration and activation of immune cells at sites of infection, as well as in cancer patients with bone metastases. IL-8 increase has also coincided with increased bone resorption. CCL20, it can be expressed in several tissues throughout the body, but in sites of injury, it's involved in the recruitment of cells needed for resolution of injury. It's also been involved in epithelial lining diseases such as irritable bowel disease, and also shown to be implicated in rheumatoid arthritis. The inflammasomes we also analyzed with no significant differences in bone, but with circulation having increased expression of NLRC5. NLRC5 has been shown to co-work with NLRP3 to induce caspase-1 activation. We've also looked at the FGF23 pathway, as FGF23 is involved in the bone, kidney, cardiac immune axes. Its receptor, FGFR1, was significantly reduced in bone, indicating a lack of ligand, a potential lack of function of FGF23 in bone, BCO-affected bone. We then took the key cytokines that were significantly upregulated in both blood and bone, or bone specifically, and challenged the HFOB cells and primary chondrocytes with the data not shown, showing that both IL-1 beta and TNF alpha were enough, treatment was enough to induce significant bone cell decreased viability. So in conclusion, BCO, we were the first to show BCO having systemic effects on pro-inflammatory factors, and that these factors form a unique and detectable signature of BCO that could contribute to the etiology, and also it gives a foundation for potential biomarkers for this BCO condition. So while all of these pathways have been shown to be involved, they all are specific. One of the major limitations for many of them is the use of HFOB cells as a human cell line. While it has shown to match bone tissue in avians, we sought to, if we could take it back to the bird and improve our in vitro model. To that end, we published in 2020, we will be finishing up publishing, but it has been accepted and is available for view, the primary growth plate chondrocyte isolation, culture, and characterization from modern broilers. So animals were hatched here at our facilities and grown to 10 days old with ad libitum access to food and water. Before chondrocyte isolation, which chondrocytes were isolated from the proximal tibial head, where a crosscut was made into the head before shavings from the growth plate were taken, placed into serum-free media before being digested. And chondrocyte cells were cultured at a density of 2 × 10⁵ cells per centimeter squared, so a high density plating. And within complete media, which had sodium pyruvate and ascorbic acid added to it in order to maintain phenotypes. We used citation 3 for live cell imaging, and we also looked at protein and RNA expressions. So I'm not the first, obviously, to isolate primary chondrocyte cells from even avian species. And however, I did seek to fully characterize these cells within my culture conditions in order to optimize them for either immortalization potentials in the future or just as a BCO model. To that end, though, chondrocyte cells in culture have been shown to often de-differentiate. So this diagram here takes chondrocytes from starting phases of the mesenchymal precursor cells down all the way through what happens within tissue, which they end up going from chondroprogenitor cells, which express collagen II, into proliferating chondrocytes, expressing collagen II and aggrecan, and then finally hypertrophic chondrocytes, with a shift from collagen II expression to collagen X, as well as other factors in the extracellular matrix. However, in culture, chondrocytes have been shown to undergo de-differentiation, which is evidenced by a shift from collagen II production into collagen I production, which is a negative chondrocyte marker, and often shape changes within culture as well. So taking these studies, we sought to identify what our cells were doing within culture at different stages in order to optimally determine the phenotype we were aiming for that best can mimic the growth plate environment under BCO and then used for BCO modeling. Starting with some live cell imagings, these are all the same magnification from days 3, 7, 11, 14, 18, and 21 in culture. So starting on day 3, most notably, you can see how densely these cells are plated. High density plating has been used for a really long time in order to create an almost 3D-like environment for these cells, which tend to respond better in that kind of environment. So they are able to form properly among one another. However, as you move further on in culture, starting at day 7 and most strongly at 7 and 11, you can see that not only does the shape change occur, where instead of rounded, smaller cells, you have flattening of the edges between cells, almost a cobblestone-like shape. But you also have reduction of that high density plating as cells will slough off within the media naturally. This shape change maintains throughout day 21. In the past, these shape changes have been used as a marker of when to use these cells as a model for growth plate diseases, when to treat them with either bacterial infection or something else within culture. And we went into this hypothesizing that this shape change would be indicative of the optimal phenotype with increased collagen II production, and that as the culture went on and the cells seemed to get larger with potential nucleus change alterations as well, that they would even enter into a hypertrophic state and start expressing collagen X. So we started looking first at gene expression. We have collagen 1A1 and collagen 1A2 here on the top, and the trend showed an increase over culture period time, showing that potentially they could be dedifferentiating over time, which was all fine at first. SOX9 is a main driver in chondrogenesis and proliferation. It was shown to be highest at early stage culture and decrease over time. Collagen 2, our main interest marker looking for proliferating or hypertrophic chondrocytes, was again highest in early culture and decreased over time. ACAN showed a spike at day 7 and then a sharp drop mRNA expression-wise, and collagen 10 again was highest at early stage and dropping over time. Looking at these factors, protein expression, both in the cell lysate on the left and in the media on the right, we can see that ACAN increased over time, with collagen 1 increasing over time as well, being highest at day 14 and 18. And this coincides with the media secretions, with collagen 1 increasing really starting at day 11 and then spiking at days 14 and 18. Collagen 2 was highest in the lysate in early culture period, starting with day 3 and day 7. In the media, day 7 showed the highest peak in collagen 2 production, with a drop after day 14. Collagen 10 significantly increased in the cell lysate over culture period. However, in the media, it was only really detectable in early stages of culture. And SOX9 had no significant differences over time. These protein expression of shifting in collagen 1 to collagen 2 was also looked at under immunofluorescence at day 7 and day 18. Day 7 was that time point in which the media had highest secretions of collagen 2. And we can see with collagen 1 being in red on the left and collagen 2 in green on the right, and then shifting from day 7 to day 18, there is an increase in that collagen 1 production. We wanted to make sure to check the functioning of these cells. So we wanted to show that collagen 2 was being produced and secreted by these cells within culture. We used a known inhibitor of vesicle secretion, brephyllin A, on cells at day 7 of culture that peak collagen 2 production. So on the left, we have the cell lysate showing collagen 2 within cells. And on the right, you can see that brephyllin treatment pretty much completely knocked out secretion of collagen 2 within the media, showing that these cells are actively producing and secreting collagen 2. So to summarize our findings, we broke up our culture periods into early, mid, and late, where our cells showed expression of collagen 2 in both the cells and media secretions in early culture period, as well as collagen 10, which was unexpected, but led us to hypothesize that due to the scraping of the growth plate, we could be digging deeper into the growth plate levels of proliferating and hypertrophic chondrocytes than we were initially thought, leading to a mix of chondrocyte cells at different stages in that early culture period. And then as we move into mid-culture, there's still some collagen 2 production. However, collagen 1 has come in both the cell and the lysate, the cell and the media. And by late culture period, with that intense cobblestone-like shape change, collagen 1 is the predominant factor being produced both in the cell and the media, showing evidence of dedifferentiated chondrocytes. So that was going through all of the molecular mechanisms found in our lab as to what could be contributing to bacterial induction of lameness and BCO-induced lameness. However, what I want to talk to you all about next is how we can take this research and bring it back to the bird. What can we really do with these findings, and what do we need to look at in the future in order to make better sense of BCO? But before that, I'm going to hand it over to Gabriel for an audience poll.
Thank you, Allison, for the great discussion so far. As Allison mentioned, before we continue, we would like to ask a couple of quick poll questions, and the questions are the following. First one is, have you used DEXA imaging in the past? The second one is, what is the main focus of your research? And the third one would be, would you like someone to reach out following the webinar to provide further information? We will wait a few moments to let everyone answer the questions, and please select the one that applies to you the most. Thank you. We'll just wait a few more seconds. Okay, thank you for answering the poll questions. I will now hand over the presentation back to Allison. Thanks.
Thanks, Gabriel. So I'd like to talk about what we can actually do about BCO. A lot of my PhD research focused on lameness and severe lesions and the molecular mechanisms behind the bacterial infection of it. But that's all fine. But what can we take back to the grower, the primary breeder, the nutritionist, and the processor? All of them care about BCO. And I would argue that they not only care about the lameness induced by BCO, but also subclinical BCO in broilers. Subclinical BCO can affect the primary breeder as allowing it to persist within our genetic lines. It can affect the grower, where birds with subclinical BCO, while not being culled for lameness, will still have decreased performance and decreased animal welfare as they're dealing with an infection. The nutritionist can target BCO subclinically as the next target for improving utilizing their feed additives or diets in order to offset subclinical BCO. And the processor cares about it as having to cull or discard leg quarters due to subclinical BCO lesions being present. But what do they care about specifically? First, they want to prevent BCO. We also want to be able to detect it. That includes both phenotyping for the primary breeder as well as diagnosing both subclinically and lameness induced. And finally, our main goal would be to be able to treat it or mitigate it if we aren't able to eradicate it from our populations completely. But how do we go about doing that? My argument is that we need to improve our mechanisms of measuring BCO. Lameness is not enough. And there's several different means of analyzing bone health as well as bacterial load on an animal. I'm going to focus on bone health. Both are involved in BCO, but I think bone health is the start. If you can have a healthy bone, you can have no wound sites. And then even if there are bacterial in circulation, they're less likely to cause an infection. There's several that have been used in the past or are used currently and have been used in the past, although most of them involved culling and necropsying and excising the bone in order to do them. I'm thinking 3-point breaking strength and classical bone mineral density analysis. The one I'm going to talk about today is DEXA, dual energy X-ray absorptiometry. It's been used more and more in the animal world, has been used heavily in human medicine and human health as a means of analyzing the bone mineral density and bone mineral content, as well as the fat to lean ratio in humans for overall health parameters and also diagnosing of bone health. The DEXA that we used has been designed for preclinical mouse models, so smaller animals with a smaller field of frame. However, what's great about it is that's completely self-contained, so self-shielding, and the screen time is extremely short. meaning that you can anesthetize animals for imaging, but you also can use ex vivo tissue or other tissues and be able to do large amounts of tissues in one day. When we actually had it here in our lab, I was able to do in the hundreds of screens in a day, just because of the screen time availability. So we sought out this means of measuring to see if we could add it to our toolkit for analyzing bone health in broilers and in our experimental research here. So when we had DEXA, what we tested was live bird imaging. We looked at ex vivo leg quarter analysis. We also looked at breast myopathies, specifically woody breast, and in ovo imaging. What we found was that we were able to look at this entire skeletal of younger birds where we would be able to then diagnose things like kinky back or severe skeletal abnormalities and image them, as well as looking at hard data from the DEXA machine, where we found bone mineral density and bone mineral content analyses to be extremely important, with even significant differences across treatment groups. I won't be showing the data here, as we hope to use it in future publications. We also were able to use the DEXA in order to image embryonic positioning in ovo before hatching. So for the live bird imaging, here I have an example. This was an anesthetized 2-week-old broiler chick, and it's laid here in 2 different positions. On the left, it was laid on its back. It was anesthetized using an injectable. And then this is the bone mineral density image from that image. What's really beautiful here is you can clearly see the spinal cord. You would be able to see if there was severe abnormalities. You also can very clearly see the proximal tibial heads. The proximal heads are a bit more difficult, but the tibias are right there, shown in this zoomed in image here as well. We tried several different positionings with the live anesthetized bird. This was it on its side. It did not take well to it being on its front, probably due to the weight and having harder difficulty breathing. But on its side, zoomed in here, we can really see that proximal tibial head again at a new angle. So we'd be able to get multiple angles of that proximal tibial head. These are those images zoomed in again with the bone mineral density shown here as well, where you'd be able to take analyses of bone mineral density and bone mineral content of the leg quarter as a whole, as well as the femur and the tibial separately, as well as even using the ROI drawings or areas of interest. You could take the proximal and distal regions specifically for bone mineral density and content. Here's some examples of the in ovo and newly hatched chick images we took. So this was a post-mortem chick, and this is in ovo. This is embryonic day 15. So not totally formed or hardened skeletal system yet, but we were able to see the beginning of the spinal cord in here and some evidence of future imaging, being able to then see positioning. And again, here with a newly hatched chick, even with the small skeletal system, we were still able to get really beautiful images of the spine as well as the leg bones. So the key takeaways I'd like for you all to take from today is that understanding the molecular mechanisms in BCO is what can provide the foundation for many things, like a non-invasive biomarker or therapeutic or selection targets for BCO. While BCO is bacterially derived, the molecular process involved point to metabolic, immune, and bone health pathways. They also with developing a reliable and relevant in vitro model for avian growth plate diseases, we could dive deeper into actual mechanistic underpinnings of these pathways in order to specify and target our therapies or selection more effectively. And that's where the primary chondrocyte model comes into play. And finally, the fast screen time and accuracy of this self-shielded small DEXA imaging platform is a promising tool in developing more robust phenotypes, not just for BCO, but overall bone health, skeletal disorders, and even in ovo imaging for analysis of in ovo health and preparation for hatch. However, I will say that streamlining the process for the DEXA will be key, streamlining both the imaging and the ROI drawing and analyses. and in general for the research is needed to fully elucidate how all of these pathways and imaging tools can be combined in order to create improved phenotypes and do something about BCO. And with that, thank you all for being here. I really hope we can have a good long Q&A session, as it is my favorite part.
Thank you, Allison, for the presentation. As Allison mentioned, we will now move on to the Q&A section, with the first question being, what future research in BCO are you most interested in pursuing?
So this research that I presented today was my PhD research. I started my postdoctoral research in July with the support of AB Vista, which is a nutritional company headquartered out of the UK. I'm really looking for means of utilizing the understandings I've developed to then offset or improve BCO instances in flocks and overall bone health. To that end, I'm investigating the use of different feed additives and inclusions in their impact on bone health, as well as microbiome cultivation, and looking at the bone microbiome, gut, bone axes. I also am really interested in improving phenotypic measurements of BCO, which is one of the main reasons I looked into the DEXA machine, and our lab sought to utilize it for our own demonstrations.
Thank you for answering the question. The second one will be, how do we best combat bone health issues in modern broilers?
So there's many different avenues you can take for bone health. There's the genetic component. The heritability of bone has been bone phenotypes have been historically pretty low. However, I think that could be a product of our means of measuring and phenotyping bone health, and specifically the fact that we do have to necropsy and cull and investigate a sibling of a bird. We can't then use it in our breeding program. Another one that I think is huge would be nutritional interventions. So enzyme inclusion in the diets, things like phytases and xylanases and xylosaccharide combos can increase gut health, so they can target the bacterial side of BCO, and that improved microbiome can also then feed the bone in improving bone health that way. And then also they can increase the absorption of nutrients needed for bone health and bone formation, as one of the key factors in BCO is bone attrition and lack of bone healing. And then there's the immune side. I think we need a holistic approach. We need to be able to look at both the bacterial and bone health side of BCO, as well as the genetic and nutritional components of the birds, and anything we can use in our repertoire and in our toolkit to improve the health of the broiler is what we need to use.
Okay, thank you for answering the question. We'll take a few moments. Remember, if anyone has a question, please feel free to input them in the Q&A section box. We will just wait a few moments. Okay, thank you then. I want to let everyone know that we have reached the end for our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any questions in the written transcript, and we will work to get this out to you over the next few days. I would like to thank Dr. Allison Ramser for the great presentation today, and I trust that we have been able to provide you with a better understanding of molecular mechanisms behind lameness in meat chickens. If in the days and weeks to come you have further questions about modalities discussed today, I encourage you. Oh, we do have a question. We just received a question. So it would be, do you think that some of your biomarkers, cytokines, chemokines, could be used for diagnostic purposes?
So that was the hope for the research into the cytokine and chemokine profile, is to lay the foundational study for being able to detect circulating biomarkers for BCO. I think they could be. I think there needs to be further research comparing these profiles in other not only bone disease such as kinky back and TD, is the profile the same or does it differ? But I also think defining these profiles over time in the broiler's life could be extremely useful as a means of potentially determining the profile that indicates progression of BCO, which is almost like a unicorn we haven't been able to get our hands on, is how do you even start to look at the progression of BCO? Therefore, I think my work shows the foundation for them being biomarkers. I think the utilization of omics techniques, high throughput, like proteomics, transcriptomics, for both the blood, the plasma, and the bone could be huge in streamlining the identification of biomarkers under BCO conditions.
Perfect, thank you. We also have another question, which is the following. You mentioned a link between intestinal microbiome and BCO. Is this correct? If so, is there a risk set of dietary ingredients or nutrients that you would avoid in order to avoid BCO, or is this yet to be determined? Allison, you can also see the questions in the Q&A section. So
yeah, I see it.
Okay.
Great question. Has yet to be determined exactly the link between intestinal microbiome and BCO. Dr. Weidman did work analyzing the microbiome of the local bone under BCO conditions, and he found what you would expect under a bacterial infection. So increased populations of the pathogenic bacteria, decreased microbiome diversity. However, no one's looked at actually connecting intestinal microbiome and BCO. The theory lies in the fact that an improved intestinal microbiome can increase tight junction function and expression, which can decrease bacterial load within circulation, which then could offset BCO conditions. And so to answer the second part, any dietary ingredients or nutrients, again, it has yet to be determined, but I hypothesize that anything that would increase inflammation of the gut would then lead to increased bacterial load within circulation and then lead to potential bacterial infection of the bone. And on the flip side of bone health, anything that increases things like short chain fatty acid production has been shown to increase bone density and bone growth and help regulate bone cell homeostasis and the ratio of bone forming to bone absorbing cells and chondrogenesis necessary for the long bone growth that occurs in a broiler's lifespan. So anything that will improve that, plus those minerals that everybody knows about, classical minerals needed for bone health, calcium, phosphorus, would all be hypothesized to improve bone health. But it has yet to be determined, but I think it's a major point of interest in seeing if it can be done.
Thank you, Allison. I will now ask the audience any final questions. Please feel free to ask them in the Q&A box. We'll just wait a few moments. Okay, in that case, as I was mentioning before, I'd like to thank Dr. Allison Ramser for the great presentation today, and I trust that we have been able to provide you with a better understanding of the molecular mechanisms behind lameness in meat chickens. If in the days and weeks to come you have further questions about the modalities discussed today, I encourage you to reach out to us here at Syntica, and we'll be happy to discuss them further. Thanks again to all of you for taking the time out of your day to attend our session, and we look forward to seeing you at a future Syntica event. Have a great rest of your day.
Thank you.